Let's go ahead and move ahead now to the next talk. This is by Aishwarya Pavithram from the University of Bergen on in vitro functional analysis of human HNF1B variants and variant interpretation and clinical diagnosis. Aishwarya.
Hi, myself Aishwarya Pavithram. So I'll quickly give a brief introduction on monogenic diabetes and focusing on especially on MODY, and will present the data obtained from our functional investigations on HNF1B variants. So to start with, maturity onset diabetes of the young, or in short MODY, is a form of monogenic diabetes which is caused due to the heterozygous mutations in genes which are very important for healthy development and functioning of pancreas. So to date, mutations in 14 different genes are known to cause MODY, including the transcription factor protein, HNF1B, as you can see from the figure. So HNF1B is responsible for causing MODY5 together with rare extrapancreatic features, especially renal cysts. Particularly in our study, we aimed to characterize the functional pathogenicity of 7 different HNF1B variants that were identified in a patient study cohort from the University of Maryland, USA. So since HNF1B is a transcription factor protein, once the constructs were made, the transactivation potential was evaluated for all variants, followed by assessing the DNA binding ability, subcellular localization, the gene and protein expression levels for each of the variants. And as recommended, we used the standardized ACMG-AMP guidelines in order to classify the variants. Here, it's important to mention that all experiments were performed in an HeLa cell line which lacks endogenous HNF1B expression. So if we look at the graphical data of all functional assays performed in HeLa cells, so out of 7 assessed variants, the A167P, which is a missense variant, demonstrated significantly reduced transactivation potential. And also it has also shown severely reduced DNA binding ability, reduced mRNA, and also severely reduced protein expression levels. On the other hand, the truncated histidine 336 frame shift variant also demonstrated severely reduced transactivation potential. However, very interestingly, the variant has shown normal DNA binding ability, normal gene and protein expression levels. So altogether, our findings allow the reclassification of 2 of the 7 identified variants. Firstly, the D82N variant. In this case, both functional evidence and also the allelic frequency data from NOMAD supported the benign evidence of D82N variant. And thus it allowed to reclassify this variant from likely benign to benign. Secondly, the A167P variant has been reclassified from uncertain significance to likely pathogenic, as the variant demonstrated severely reduced functional defects in our studies. Also, the frame shift variant, which was previously classified as pathogenic based on the clinical feature of the patient, as the patient is born with 1 single kidney, a feature which is consistent with MODY5. Indeed, our studies has also shown significantly reduced functional activity and thus supported its pathogenicity. So finally, the remaining 4 variants demonstrated near to normal HNF1B activity, and thus we could not really conclude much on these variants, and they still remain classified as VIUS. Hopefully, to conclude, our studies underscores the importance of functional evidence in the reclassification of VIUS variants, and we hope a study would add value to the affected subjects and their families, leading to a better diagnosis and treatment. Thank you.
Okay, thank you very much. If you could drop your slide set, thank you. No questions have been posed, but I might ask about the management of these patients with the various types of genetic mutations in terms of the phenotype that these patients present with. I think you showed the renal agenesis phenotype was one, which was associated with one of the variants. How about the others that were associated with type 2 diabetes? Do we have any information about the patients who had those?
We really could not see any phenotypic features that are associated with MODY5, but it would be really interesting to get like more clinical data, and that's what we are exactly waiting for. And it could be that they are not associated with like HNF1B, like mutations might not be pathogenic, but I'm sure that if we investigate more, there could be the other pathogenic variants that are found on other monogenic genes, which is causing diabetes to those patients.
All right. Well, thank you for very nice work in presenting today, Aishwarya.